Hi everyone, welcome and thanks for joining us today. My name is Laura MacDonald and I am the Knowledge and Research Coordinator here at CAWST. We're joined today by Han-Ting Wang and Helen Nguyen from the University of Illinois at Urbana-Champaign. And Han-Ting is a master's student whose research focuses on virus removal by biosand filters. And Helen is an associate professor who's an expert on biological contaminants in the subsurface environments and on water treatment systems. And I want to say thanks for joining us, Han-Ting and Helen, and if you're able to, could you turn on your webcam and say hello?
Hi.
Hi. Thanks, Helen and Han-Ting. All right, so now we'll go on to the presentations. I will be giving a brief introduction, and it'll just, so y'all can turn off your webcams now. Thanks. All right. So after a brief introduction, I'll hand things over to Han-Ting to present her research on virus removal in the Biosand filter. And then from there, we'll address questions that we gathered from US participants during the registration process. And then throughout this time, Emily will be keeping track of comments and questions that you contribute through the chat function on GoToMeeting. And so after the pre-submitted questions are discussed, we'll take some time to address several of these new questions. And for the questions that aren't answered today, there will be a follow-up email after this webinar. And also, as a note, we are recording the webinar, so that will be made available afterwards as well. And so, as for what's going to be presented today, when the work by Han-Ting and others was published last year, we were excited to see new information coming out about virus removal, particularly because for a long time, little has been understood about the mechanisms of virus removal in the biosand filter and factors that influence this removal. And some believe that the biosand filter doesn't perform well with respect to virus removal, but this research shows otherwise. And at CAWST, we want to learn more and would like to know if high virus removal is achieved by other biosand filters. And we'd also like to consider how the design of the filter could be changed to achieve these high levels of removal. And so when I say we, I mean the Research Learning Department at CAWST, because one of our roles within CAWST is to stay on top of emerging research that's relevant to the work that we do on water sanitation and hygiene, with biosand filters being a long-standing interest and focus within the organization. And to do that, we stay in touch with researchers at academic institutions to learn about what they are doing, and then serve as a link to implementers and facilitate collaborations between researchers and implementers. But as I said, we're interested in this research because of its relevance to what CAWST does. And so it's also our job to communicate important findings to the other departments within CAWST. And so when this research came out, we sought out the opportunity to have Han-Ting and Helen share their findings with our staff. But we quickly realized the opportunity we had to share these findings with a much broader community. And given that biosand filters have been implemented in 62 countries, there is truly a massive community to whom Han-Ting's research is relevant. And we're so glad that many of you could join us today to learn alongside our staff. And so with that, I will hand things over to Han-Ting for her presentation. So if you have questions as they come up, just feel free to enter them into the chat, and Emily will be keeping track of them. And with that, I'll hand it over to Han-Ting. Thanks.
As Laura mentioned, I'm a master's student at the University of Illinois, and I'm joined here by Professor Helen Nguyen, who's an associate professor in the Department of Environmental Engineering. So here's an overview of what I'll be presenting on today. First, I'll be discussing some background on point-of-use treatments, biosand filters, and then viruses. And I'll move into some previous experiments and motivation for our study that we just published on. And then I'll dive into MS2 removal due to physical filter factors and then microbial communities. And I'll finish off with some implications of the work and some future work. So the World Health Organization estimates that 780 million people around the world still lack access to improved drinking water sources. Many of these people live in impoverished communities where children are most affected by diarrheal diseases. In these areas, typically a centralized water treatment system is not feasible due to cost and land restrictions. However, recent studies have shown that point-of-use systems in which water is treated at the location of consumption, are able to reduce diarrheal diseases by up to 40%. Here are some examples of point-of-use systems that are popular. On the left, solar disinfection. There's also flocculation and chlorine tablets, ceramic pot filters, and then on the right side is a Biosand filter. A study by Professor Mark Sabzi from UNC Chapel Hill in 2008 showed that Biosand filters are the most promising point-of-use system currently available. Since 1990, over 770,000 Biosand filters have been implemented in more than 60 countries around the world, as you can see in the map on the top left. These filters are easy to use, widely accepted, cheap, and marketable. And studies have shown that they're able to remove bacteria and turbidity well. Also, the average filter is about $40 US, and this cost includes labor, materials, and transportation. Biosand filters are intermittently operated around every 12 to 24 hours, and I have a schematic here of how the filter works. So first, one pours in 12 liters of dirty water into the filter at the top, and the filter works through gravity, so no extra energy is needed. And so you can see that the water flows down through the filter, and we let this water sit for 12 to 24 hours, and in that time it becomes filtered. And then you pour in another 12 liters of water, and that pushes the clean water out into the covered container. And here I show this green layer at the top, which we call the biolayer or the Schmutzstecke, and I will talk more in depth about that later on in this presentation. So before I move into previous studies, I want to explain a little bit about why we're looking at virus removal. First, it's because, in general, viruses are hard to remove through sand. So as you can see on the left side, we have yellow sand particles and blue virus particles. And in environmental conditions, both viruses and sand are negatively charged, and so there's natural repulsion between the two. On the right side, I show that bacteria, which are about 100 times bigger than viruses, are more easy to remove physically than viruses because of the size, viruses are able to pass through pores between the sand particles. Here I show some data from Professor Mark Elliott's work from 2008 comparing bacteria and virus removal in biosand filters. On the left is E. coli removal, and on the right, Professor Elliott compares the removal of 3 different viruses. The x-axes are days of the experiments, and the y-axis is log reduction, and this is a metric that's used to express levels of decreased biological contamination in water. And so 1 log removal is 90% removal, 2 log is 99%, 3 log is 99.9%, and 4 log is 99.99% removal. And 4 log is the US EPA standard for virus removal. So on the left, we can see that E. coli can be effectively removed up to 4 log in just 50 days. But on the right side, note that I highlighted that the y-axis only goes up to 2.5 log. And we see that only after 50 days do we even see a little bit of increase in virus removal, and only for one of the viruses. The other two, MS2 and PRD1, don't even hit 1 log. And so because of this, we want to focus more on virus removal than bacteria removal. The last thing I want to talk about before getting into our experiments is why we chose to study MS2. First, MS2 is a good surrogate for human enteric viruses. It is small in size, so about 30 nanometers, which is relatively small even among viruses. So being able to remove MS2 effectively could indicate that larger viruses could also be removed. Also, MS2 has similar morphology as enteric viruses. And finally, MS2 is easy to work with compared to other viruses. Propagation and quantification methods have been well developed and used. So here's a figure of biosand filters that we constructed and experimented on at the University of Illinois. On the left, there's 3 version 9 filters, which have 45 centimeter sand heights and 20 liter charge volumes. And on the right, we see the newer design of the Biosand filter, the version 10 filter, which has a 55 centimeter stand height and a 12-liter charge volume. And so we can hypothesize that the version 10 filter, which is taller and has less water fed into the filter at a time, should get better removal than for the version 9 filter because there's more contact between the water and the sand. And this is exactly what we saw in our experiments. So once again, the y-axis is log reduction of MS2, and the x-axis is days of experiments. And I also drew a line at 4 log removal, which shows the EPA standard. And we can see that for the version 9 filter, which I indicate in black, that only after 200 days do we see the removal reach the EPA standard of 4 log. And even after that, it's not very consistent. However, for the red, blue, and green lines, we can see that for version 10 filters, the removal reaches the EPA standard pretty quickly and consistently. And so this leads to the objective of our newest study, which was to try and determine the mechanism of MS2 removal in biosand filters. And while several previous studies have suggested possible roles of physical factors and biological activities within the filter, the individual and synergistic roles, contributions still remain uncertain. And here, when I say physical filter factors, I mean filter depth and residence time of the water that sits in the filter. And for biological activities, that includes microbial communities that develop throughout the filter with time. And this study is the first to use genomic sequencing techniques to determine microbial communities in the biosand filter. And improved knowledge of the mechanism and magnitude of virus removal can be used to enhance the design and efficiency of future biosand filters, which could lead to improved performance and reduced disease incidence. And so, to better understand the mechanism of virus removal in the biosand filter, we built a port filter shown on the right. It's the same thing as a normal version 10 biosand filter, but we inserted 7 ports down the depth of the filter so we could take water samples from each port and eventually core sand samples. For 240 days, we took water samples to determine the effect of depth, residence time, and media aging or ripening on MS2 removal. After that, we took core sand samples at each depth and analyzed concentrations of protein and carbohydrate present in the sand samples. And then finally, we performed 16S rRNA sequencing to determine which bacteria species are present throughout the filter at different depths. One of the most important findings in the study is that filter ripening is essential for high removal viruses. There are 2 types of ripening that occurs in the filter. The first one is that biofilms develop around individual sand particles throughout the filter. On the left, we see water flowing through with other particles through the sand. And on the right side, we see that as more water and particles flow through the sand, there will be biofilm, which I depict in green, that builds up around the sand particles that constrict pore spaces and slows down the flow of the water. A slower flow rate means that there's more contact time between the water and sand particles and overall higher removal, similar to what I mentioned earlier when comparing version 9 and version 10 filters. And we can see just how much ripening improves MS2 removal in this figure. This is cumulative log 10 reduction of MS2 as a function of depth and as a function of media aging. So before I go into this data, I just want to explain a little bit about how the experiments worked. So for this figure, we used 2 different filters, one that was for days 9 to 27 and one that was for days 43 to 240. And for the days 9 to 27, we called that a non-ripened filter, which is a scaled-down version of the version 10 Biosand filter, and it had a 2.1 liter charge volume. So when we ran the filters, we would spike MS2 into our feed water and pour that into the filter. After the water finished running through the filter, we would collect samples at each port, which we denoted as short residence time, because the water only had about 10 minutes of time for of contact with the sand for the unripened column, and then for the ripened column, which is days 43 to 240, it had about a 45-minute short residence time period because of a larger charge volume. And then before we poured in water again the next day, we took samples from each port again and denoted these as long residence time because the water had around 24 hours of contact time with the sand. And so for this figure, this is for short residence time only. And we can see that before 28 days, there's pretty low removal, only up to about 2 log with short residence time. And the removal in the top half of the filter is pretty negligible, and only after a depth of about 32 centimeters do we start to see some increase in removal. However, as we continue to run the filter for more days, we see that the cumulative MS2 removal increases to more than 4 log, and this is only with short residence time of about 45 minutes. Also, a recent study done in our lab group showed that using the same groundwater source we use in these biosand filters, biofilm develops fully after around 4 weeks, and so this matches our hypothesis that ripening is very important for removal. The second type of ripening that occurs in the filter is where biomass accumulates densely in the biolayer due to extensive biofilm growth and the lack of backwashing. This type of ripening is really crucial for the efficiency of the filter, and we can see that through the figure on the right. So once again, this is cumulative average log reduction of MS2 versus depth and as a function of residence time. And this filter, which we say was with ripening, was run up to 240 days. And what we can see here in the blue with long residence time of about 24 hours, we see that there's 3 log removal just in that 5 centimeters alone, which is the biolayer. And although the cumulative reduction increases up to 5 log, we see that the rate decreases with depth. And we see a similar trend with short residence time where almost 2 log removal is seen in the first port, and then the cumulative reduction increases, but at a slower rate. And so what we say here is that most of the removal takes place at the top of the filter, and so the biolayer is extremely important. Another important thing to note is that physical characteristics like depth and residence time are critical factors when the filter is unripened, meaning the biofilm is not fully developed. And we can see this through the 2 plots that we have here. So for a non-ripened filter, we see that there's an exponential dependence on reduction, and that and compared to a ripened filter where the highest reduction takes place in the biolayer. What we see for non-ripened filter is that if you don't have a filter that's at least 45 centimeters, then you will not be able to reach 4 log removal. And so the depth of the filter is extremely important. Another thing to note is that residence time is really important, more important for a non-ripened filter than for a ripened filter, because if you don't have residence time of about 24 hours, you will also not be able to reach the 4 log removal. So after we finished our MS2 removal experiments, we took core sand samples throughout the filter by sticking a 1-inch PVC tube into the filter and collecting sand at different depths. We used some of the sand to extract proteins and carbohydrates, which are the main components of EPS, which is a sticky substance that bacteria excrete when producing biofilm. Here, what we saw was that there was not significantly different concentrations of proteins throughout the filter, but there was a significant difference in carbohydrate concentrations in the biolayer compared to the rest of the filter. And we did see that there's activity of carbohydrate degrading bacteria throughout the filter, and so this depth-dependent distribution of carbohydrate may be related to that. So the last objective in our study was to determine what bacteria species are present in the filter and try to correlate that with MS2 removal. So to do this, we took more of the core sand samples that we took, And we extracted DNA from the sand, performed PCR and gel electrophoresis, purified the gel, and quantified the DNA concentrations before sending them to a center on campus for sequencing. So here we show 4 alpha diversity indexes, which depend on the number of species present in the sample and also the proportion of the species represented within the sample. And so a community with a high alpha diversity will have a high number of species and high abundance. High number of species and abundances will be similar, I'm sorry. And so what we can see here through all 4 of these plots is that there's a higher microbial diversity at the top of the filter, and this diversity decreases with depth. We also looked at how microbial community structures shifted with depth, and this is through a principal coordinate analysis. And we can see that there's a clear shift in microbial communities among the samples that were taken throughout the filter. And the shift of microbial communities and the decrease in diversity with depth shown in the previous slide correlate with our MS2 removal data, showing that a lot of the removal takes place in the biolayer and the rate decreases with depth. Here's the diagram that shows the relative abundances of classes of bacteria as a function of depth of the filter. And I just want to note here that the main 2 classes of bacteria that dominated are in the red and the green, so beta proteobacteria and gamma proteobacteria. And one of the most important observations in our sequencing analysis is the detection of chemolithotrophs that were found throughout the entire filter. In the lower depths of the filter, we saw Biobacillus, which is a sulfur cycling microbe, and those dominated there. And in the top parts of the filter, we saw Candidiasis nitrosopamillus, which is an ammonia oxidizing archaea, and also a Nitrospira, which is a nitrite oxidizing bacteria. A couple previous studies on microbial community structures of large-scale sand filter processes treating groundwater also showed significant populations of AOAs. And also a recent study showed that nitrification and denitrification were observed in biosand filters operating in Cambodia. So taken together, these chemolithotrophs that dominate throughout the filter might play an important role in the reduction of MS2, but the function of these chemolithotrophs on MS2 reduction is still unclear, and further studies need to clarify the functions of these chemolithotrophs in biosand filters. So in conclusion, our study was able to show that cumulative MS2 reduction in biosand filter can reach EPA standards, and for us it reached an average of 5.6 log after 240 days. What we know is that MS2 reduction in a biosand filter depends on physical characteristics for unripened filters and microbial communities for ripened filters. We also saw that that 16S rRNA pyrosequencing analysis showed that microbial compositions of biosand filters change significantly with depth, and that various types of chemolithotrophs might play an important role in MS2 reduction, but that needs to be further studied. Finally, the development of biolayer is extremely crucial for increasing MS2 reduction, and that leads to my last slide, which talks about the maintenance of the biosand filter. So on the left is this handout that we developed through CAWST materials for a biosand filter project in Guatemala. And this basically shows the swirl and dump method, which is used for maintenance when the filter starts to clog and the flow rate slows down. We tell people to swirl the top few centimeters of the filter. And while this helps to unclog the filter, this disrupts the biolayer. And there are some previous studies that have suggested that since biofilm develops throughout the entire filter, perhaps there isn't an issue with disrupting the biolayer. However, our study showed just how important the biolayer is, and so what we would like to do is to determine how to enhance biofilm development throughout the entire filter. If we could somehow design the filter so that the communities that take place in the biolayer develop throughout the entire filter, then when maintenance is needed, the removal efficiency won't be compromised. So that's something that, that we believe should be studied. So with that, I'd like to acknowledge the US EPA and NSF for funding our work. Also, our UIUC Engineers Without Borders team that helped with the research, and also CAWST for their constant support and for allowing us to present our work today. So with that, I'd like to thank you for your attention, and I'm happy to take any questions.
All right, thanks so much, Hunting. And so with that, we will go into some of the questions that were submitted beforehand. And then Emily, as I said, is taking note of questions as you enter them into the chat. And any questions that aren't answered today, we will follow up on. So to start, there were quite a few questions submitted about the performance of the Biosand filter, not only with respect to viruses, but also the other microbiological contaminants. So could you touch on that, and then how the Biosand's performance in that regard compares to other types of household water treatment?
Sure. So your first question is comparing virus removal to other pathogens?
Yeah.
Great. Okay. So most of the studies that have been performed are on bacteria removal, so E. coli mainly. There's also studies done on coliform removals for in-field studies. So I think a lot of the bacteria removal studies show similar results. So between 2 to 4 log removal, which is pretty high. For coliforms, a lot of times we are able to see that there are no total coliforms left, which is, it's good, but it's not exactly an indicator of pathogens that could harm humans. And so there haven't been too many studies done on viruses, mainly just bacteriophages, and we do see a large variety of removals. So between less than 1 log for some studies and up to 5 log for us. And so there's just a huge range of removal for viruses. There's also not been any studies done on human enteric viruses, and so that's something that needs to be done as well in order to better understand the Biosand filter.
Okay. Great. And then how does the performance of the Biosand filter across the microbiological contaminants compare with other types of household water treatment?
Sure. So, for example, the ones that I showed, so for solar disinfection, given the thermal and photooxidative capabilities, I believe that pathogens are well removed, but the limitation is time and the volume of water that can be treated. This is similar for the chlorine tablets. Okay, this is similar to the flocculation and chlorine tablets, which are effective in removing pathogens, but there could be a taste issue. It's also more costly in the end, not as sustainable as Biosand filter, for example. And so while they might be able to remove more pathogens, there's other factors that need to be taken into consideration.
Great. And you touched a bit at the end about the issue of the Biosand filter or the biolayer being reestablished after the swirl and dump method. And I guess one thing to clarify to everyone is that although the swirl and dump method does disturb the biolayer to a certain extent, it's just agitating the very top of the surface dust and not digging into the sand. So we just wanted to make sure that everyone not to dig into the sand during the swirl and dump. But with regards to future research on changing the design of the filter to try to encourage growth throughout the filter, does your research have any other implications for Biosand filter design?
Well, so what we do know is that the depth is really important. So I mean, it is possible to still reach pore log removal even with a 45 centimeter filter, but we wouldn't discourage the current design, which is 55 centimeters. We also know that residence time is extremely important, so that's something that should be continued with education when we're teaching new users how to use the filter. Make sure that residence time is there. And I think that with particular water sources, oh, that's something I forgot to mention, is that our study was with one groundwater source that's been well characterized, but that's not necessarily representative of all water sources that are used around the world. And so it could work with our water source, but it may not work with other water sources. And so that's one of the reasons why we need to continue looking at the microbial communities throughout different filters, so that we can understand what's dominating, and then we can try to redesign the filter that way.
Okay, great. And another question is, you touched on the different ripening periods and what you, how long you waited in your studies, but have you any conclusions about how long one should wait after installation before using water from the filter?
Sure. So from our study, we saw that we could reach the EPA standard after 28 days, so about 4 weeks, which is still a long time. And so we do understand that users could become frustrated if they're still getting sick even after 4 weeks. So that's another reason why we want to try to develop that biofilm earlier, or throughout the filter earlier, so that we would be able to reach EPA standard earlier. But we are also the only study that has shown that we can reach EPA standard that quickly, and so more studies need to be done in order to make sure that it's possible with different sources.
Right. Okay. And then as to kind of a more technical side of things, you touched on testing for viruses in the lab. Some people wanted to know a little more about testing for viruses in the lab, but also in the field.
Sure. So it's definitely easier in the lab.
Right.
So there's different methods. So for MS2, we use plating. That's counting plaques. So it's a little more simple. We can also do PCR, which we didn't have to do for MS2 since we could have the plating method. But it's definitely more standardized and idealized in the lab. In the field, I guess the main reason why there aren't virus field studies is because it's quite difficult to quantify viruses in the field. A lot of people just rely on coliform plates, but it is possible to save the to like extract the RNA from viruses and then ship that to a lab. But that's quite expensive, quite time-consuming, and pretty difficult to do. So I think this is the main reason why there's a lack of studies on virus removal in the field. It's just really difficult to perform.
Okay. And then another question regarding viruses that we just received is, how does MS2 compare with rotavirus specifically? Is it similar to rotavirus, or?
So morphology-wise, it's similar, but it's quite a bit smaller, like 3 or 4 times smaller. And so that is one of the reasons why we used MS2, though. It's just because of the size. It's relatively small. So in that sense, it could be a good indicator for rotavirus. So our group has actually started doing rotavirus removal experiments. Period, we are able to reach EPA standards. This is using the same sand source and the same water source that we use for our experiments, though. So once again, it is, dependent. So what we can say is that it's really dependent on the water source. So the microbial communities that develop. We don't have that data published yet, so we didn't show it, but that is something that we've started to look at.
Okay, great. And then talking a little more specifically about sand, there was a question about how or if sand size influences virus removal. You touched a bit on the mechanisms of virus removal. There's the sieve that's used to prepare the sand influence virus removal at all?
Okay, so I would say yes, but we do use a specific sand size in these filters, um, so that the flow rate is not too fast or it's not too slow, but also has enough contact time. And so we've only tested using the CAWST recommendation of sand sizes.
Okay, great. And besides depth, what physical characteristics could improve the biosand filter function?
Um, Something that we tested for years to make sure that our removal of viruses isn't too strictly to, for example, metals that are part of the sand. But there are definitely sand sources that may have that. And so when those metals get oxidized, then it could potentially improve virus removal.
Okay.
But really what we see is that since we saw that our sand source did not have those metals, we could basically just say that it's depth and residence time that really play a role. And I guess we do understand that since we ran our filters every 24 hours, it's not exactly representative of those in the field that have to run the filters more. So that's something that we can also potentially look at in the future. Is like, what if we ran it every 8 or 12 hours? Do we still see the same thing? Yeah.
Okay. All right. Yeah, because Candace did, her study looked at even longer residence times of 2 to 3 days.
Right.
So,
Mm-hmm.
And then another question is, would your proposal to try to enhance biofilm growth throughout the filter, would that cause clogging further down in the filter? And how might that affect flow rate?
Yeah, that's a good question. Well, what we could also try to do, yes, that's a good point. What I was actually envisioning is that whatever species that dominate in the biolayer, like particular ones, we could potentially spike nutrients in throughout the filter so that those grow throughout the filter, so not necessarily as densely, but as long as they dominate throughout the filter, then that would work. But yeah, we would have to be careful just to make sure that the biofilm doesn't develop as densely as in the biolayer. But in general, it wouldn't, just because there's so much more going on at the top of the filter compared to the bottom of the filter. A lot of microorganisms are getting removed at the top of the filter, which is why it's so much denser at that layer.
So I just want to mention something, that what we would like to do is just encourage the species, the microbial species that help with the removal. It's not necessary that you need like very dense biofilm, just the good one.
Yeah. Right. Yeah. So the ones that are actually helping with removal would be the ones that we would want throughout the entire filter.
Okay. And as it stands currently, what are the limiting factors, as you understand it, to that growth further down in the filter?
I guess the biggest limitation is that we've only ever been able to look at one filter and one water source, and with different water sources, different microbial communities are going to develop. And so right now we can conclude things about our particular water source and filter, but we can't go on and redesign anything based on that just because it's not representative. And so more studies need to be done. And I think there are some recently published papers that are starting to look at microbial communities, but I think there's going to need to be more and more done in order to understand which communities are dominating.
Right. Okay. And okay. Sorry, there's a more involved question in the chat that I need to look at. Marion's question. Where is it? Okay. So Marion says MS2 is chosen because it's small, and if it's removed, the chances are that the bigger viruses could be removed too. But you also said that the physical characteristics are important, namely size, and as is microbiological. So this could mean that MS2 is well removed, but the other ones could not be removed due to the unadapted bacteria. So that was a comment from Marion. I don't know if you wanted to respond to that at all, of just MS2 as a surrogate and potential limitations of MS2 as compared to others.
Yes, so that is a good point. That was a generalization that I made, that due to smaller size, perhaps a bigger virus could be removed better. I understand that that may not necessarily be true. It could depend on the morphology of the virus, or if, I guess, let me think. Well, I guess one thing about MS2 as well is that it has a pretty low isoelectric point compared to other viruses in environmental conditions, and so it takes more in order for the MS2 to get removed.
Okay, so
yeah,
a lower isoelectric point, that means essentially it's more negative, or can you explain that in a...
Yeah, it's more negatively charged compared to other viruses, and so that may mean that it's even more difficult to remove than, say, rotavirus, which has a little bit higher isoelectric point.
Okay.
But also, what's been studied has only been bacteriophages, so it's really hard to generalize to all viruses. But yeah, like I said, we are starting to look at human enteric viruses. Yeah, so I mean, she does have a good point. We can't say that it'll work for everything, but MS2 is in general a good surrogate for enteric viruses.
Okay. And when you do, now that, say, you said you have started some human enteric virus research, how does the protocol change, I guess, besides being more careful?
Right. So one of the main reasons why we constructed that smaller scale Biosand filter, so we constructed it so it's only 2.1 liters of charge volume instead of the normal 12, is because enteric viruses are going to be a lot harder to work with. We can't grow it to a high enough concentration where we would be able to see effectively how much the removal is. And so with a smaller charge volume, we can spike in less virus and still perform these experiments. But we do the same thing. We spike in the filter, or spike the virus in the filter with the water. We take samples at different depths. For rotavirus, the quantification is a little more intense, where we have to do both the plating method and qPCR in order to really understand what the removal is. So it's just longer and more time consuming. But the actual experiment of running the filter and taking samples, that's all the same. We wanted to make sure that that was the same. It's just the quantifying that's a little more challenging.
Okay. Okay, and Candace had another experimental design question, and that was, how did you build ports into the concrete filter?
Yes. So this was when we were actually constructing the filter. What we did was we let me try to think. Well, we stuck okay, so when we were actually making the filter, so before it dried, we stuck in something to make the holes, and then we used needles in order to take samples. So we used a syringe and needles to take samples. I'm sorry, I'm not sure if I was there exactly when we did it for the concrete Biosand filter, but then for the PVC one, which I made, we drilled holes into the PVC. So it was easier since we didn't have to let it dry. Yeah, but essentially what we did was we wanted to make sure that we were taking samples from the middle of the filter, so our needle was long enough to reach the middle of the filter at each step. Yeah.
Okay. I believe those are all the questions that we'll address here. There have been some other questions that we'll definitely follow up with.
Okay.
And so, as I mentioned, let's see. The webinar has been recorded, so the recording will be available on our resources website. We'll also send out an email with a link to that to all the registrants. And Emily will be sending out emails regarding specific questions that weren't answered today, particularly those that were asked that were more country-specific or project-specific, and we'll help you get in touch with our international technical advisors that work in your area. And so with that, Helen and Hunting, thanks so much for joining us. Please feel free to send any follow-up questions that you have, and we'll get in touch with Hunting and Helen and get answers to you. So I hope everyone has a good day, and thanks again for joining us.
Thank you.
Thank you. Bye-bye.